Hi everyone, we are from University of Nottingham, Malaysia campus, Year 2 Chemical Engineering Group 17. Today, we are presenting you an ISE lab experiment, the determination of fluoride concentration in water. Before going on any further discussions, let me introduce you to my group members. This is me, and here are my teammates, Yashi and Jayla. Before conducting the experiment, we need to know what is ISE. ISE stands for ion selective electrode. The ion selective electrode works based on the principle of galvanic cell, which consists of a source electrode, a discriminator electrode, a sample, a detector, and an output. The ion selective electrode measures the potential difference created from the transport of particular free ions in the solution down the concentration gradient across the membrane. The potential measure is then compared with the constant potential in the reference electrode to determine the net charge. The strength of the net charge measure is directly proportional to the concentration of the particular ion. Now, you might be asking, since the electrode is measuring the activity of particular ions, why is it called ion selective electrode rather than ion specific electrode? The answer is, the electrode is still experiencing interference from the other ions. After a short explanation of the ion selective electrode, now we move back to the topic, the determination of fluoride concentration in water. The purpose of conducting this experiment is to construct a calibration data of the standard fluoride solutions. Next, to determine the fluoride concentration of different water samples and to analyze the effect of temperature variation on the conductivity of the water samples. In the experiment, the water samples given are the UNMC lake water, the tap water, and the deionized water. In the experiment, instead of using a double electrode galvanic cell, we are using a combination fluoride ion electrode, in which the reference electrode and the ion selective electrode are built in one.
Here we have the main equipment of our experiment. The equipment actually consists of three main parts. The first is the power display. Second is the conductivity sensor. And finally, we have the ion selective electrode. Now, each of these parts of the equipment have different roles to play. Let's start from the ion selective electrode. The ion selective electrode helps us to measure or calibrate during the experiment. And it also helps us to measure the fluoride ion concentration during the experiment for the different samples. The conductivity sensor, as the name suggests, helps us to measure the conductivity of the different samples at different temperatures. Finally, the display helps us to obtain the values or the numbers for the concentration using the ion selective electrode and the conductivity of the sample liquid using the conductivity sensor. Thank you very much.
Thank you, Jaber, for the presentation. Now, I'll be giving you a quick walkthrough on how is the experiment constructed. First, we prepare the solutions with fluoride concentration of 1 ppm, 10 ppm, and 100 ppm from a 1000 ppm solution. Next, 2 ml of T-sub is added into each of the standard solutions. The calibration is then carried out in a continuous sequence with increasing concentration. After data calibration, analysis of fluoride concentration in different water samples is performed using the combination fluoride ion electrode. The last step would be the conductivity measurement of the water samples at two different temperatures, the ambient temperature and the refrigerated temperature.
Next, we will talk about the results we obtained from the experiment. Table 1 shows the concentration and signal of the standard solution we prepared. We are using the data from the table to plot the calibration curve. And this graph is the calibration curve that has been plotted. From the graph, we can clearly see that there are two different slopes which are going to further discuss later on. Now, the table above shows the average concentration of the fluoride ions of each of the samples A, B, and C. The table shows that sample A has the highest average concentration, which is 0.4335, and sample C has the lowest concentration, which is 0.122.
Further on, the conductivity of each samples in ambient and refrigerator state are being shown in this table. From the table, the samples in the ambient state has higher conductivity compared to the refrigerator state. This is because the conductivity of water is temperature dependent. The conductivity will increase about 2% when the temperature rises by 1 degree Celsius. Sample C has the lowest conductivity in refrigerator state compared to other samples. Thus, from the data above, we can conclude that A is the tap water, B is the lake water, and C is the deionized water. Now we are going to talk about the discussion part. According to theory, we have two calibration curves, segmental method and linear method. Segmental method is the method that permits precise and accurate determination of the correction data for low concentration solution. More calibration are being done in this stage. And this method is connect the individual point instead of drawing a straight line that cut across the points. So it is only linear between points to points, but not necessarily across all the data points. And hence, this is what we call segmental method. Linear method is a method that finds the best fit line between the points. But in the low concentration region, the calibration method can be subjected to be error because there is a derivative in the low concentration region. So from this graph, we can clearly see that what is the difference between segmental method and linear method. The red line represents the segmental method and the blue line represents the linear method. The only difference between these two methods is the ways to connect the data. Segmental method is being used in a lower concentration region, but the linear method is being used in a higher concentration solution. One of the factors that we should consider about in choosing the calibration method is the range of the concentration of the solution. It is because in order to get an accurate result, segmental method is suitable to be used in the low concentration solution. However, linear method is suitable to be used in high concentration solution. So now we are going to compare the calibration curve that we obtained from the Excel and the calibration curve that we obtained from ISE machine. There is a difference between these two figures. It can be seen by using the extract from linearity. The calibration curve that we obtained from the machine was using the segmental method. However, the calibration curve by using Excel was using the linear method. Thus, the half of the calibration curve is not as linear as the graph in the Excel. T-sub, which is also known as total ionic strength adjustment buffer. It is used to bring out the ionic strength of the samples and standards. And also, it is used to make sure that there is a constant background ionic strength. T-sub also serves to adjust the ionic strength and to reduce interference or reaction from other ions that might possibly react with the fluoride ion to form a compound. For example, aluminium ion, iron ion, and so on. So, what will happen if T-sub is added in the inappropriate amount? First, the background ionic strength will not be constant, and this will directly affect the results obtained. Besides, some ions will react with the fluoride ions to form another compound.
From the respective concentrations of fluoride ions determined in our samples through the experiment, the confidence interval was calculated for us to use as a statistical analysis to support our argument. In addition, this helped us to compare our results with literature values against the experimental values. In order to calculate the confidence interval of the experiment, we use equation 1. X bar represents the sample mean concentration. Z star is the Z value for the desired confidence level. N represents the sample size. And sigma is the population standard deviation given by the equation number 2. Having determined the value of sigma, we can then substitute it in equation 1. This gives us the confidence interval. The sample size taken from the experiment is 2, and the confidence level is 95%. This gives us the confidence interval for the experiment. This table represents the confidence limits obtained for different concentrations of the samples. During the experiment, there were also discrepancies obtained in the experimental values against the literature values found. For sample A, which is tap water, the discrepancy could be explained by the difference in location. The tap water concentration for fluoride ions could differ from state to state. The discrepancy in sample B, which is the lake water, could be due to the fact that lakes could be used for different purposes. Lakes that are used for fishing have different ion concentration as opposed to those that are used for bathing. Finally, for deionized water, which is sample C, ideally the concentration would be 0 because, as the name suggests, it is completely treated to remove all the ions. However, due to some impurities that might have been introduced during the experiment, there is a certain value obtained for its experimental counterpart. Now, there are other possible methods to measure the fluoride ion concentration in water from different sources. Gas chromatography. In this method, extraction of the ions is achieved using trimethylchlorosilane in toluene. This produces another chemical called trimethylfluorosilane, TMFS. The organic layer is then injected into the gas chromatography system. Then the TMFS peak height is compared with those of standard solutions to obtain the concentration of fluoride ions. Colorimetry. This method is based on the bleaching of colored complexes with organic dyes. The degree of bleaching is determined with a spectrophotometer. The concentration of fluoride ions is assessed by comparison with standard solutions. Ion chromatography. This method utilizes anion exchange resins as a stationary phase to separate the fluoride ions. Conductivity detectors are then used to detect the ions in the element. It is generally applied to the separation of weak organic acids. Now, there could be a number of factors in determining the most suitable analytical method to measure the concentration. However, in our case for the experiment, ion selective electrode happens to best suit the purpose. This is because it is simple to perform, has good precision and sensitivity, it offers an economic and inexpensive method, and finally, it's widely used in the environmental media. This means that it best functions for our experiment. Conductivity and temperature variation. The conductivity of the samples were measured at two temperatures, the ambient and refrigerated. As can be observed, the conductivity for every sample was higher for ambient as compared to refrigerated. The Nernst equation shows that the total potential depends on temperature and the logarithm of ion activity. Higher the temperature and higher the activity of the ions, higher is the potential. This reflects on the fact that the conductivity would be higher and vice versa. In real-life situations, the potential would vary based on the temperature, giving us different values for the concentration measured. In conclusion, the following aims of the experiment were met: calibration, determining the fluoride ion concentrations, and comparing the conductivity of water samples. There were many discrepancies and errors involved during the experiment. Random errors could have been introduced during the measuring process for the volumes in pipette during dilution, or parallax errors while reading the volumes in the tubes themselves. Systematic errors could have happened during the instrument calibration process, or even in inaccurate concentrations being carried forward from the first process of dilution to the subsequent processes and transfer of liquids. Here are the references of the citations made throughout the video. Thank you very much for watching.